In an epoch-making endeavor, an illustrious consortium of genomic erudites elucidates the enigmatic genetic tapestry woven into the annals of Canis lupus familiaris. This groundbreaking compendium, harnessing an unparalleled genomic databank, promises to redefine our cognizance of canine evolutionary pathways and their multifarious genetic underpinnings. Despite the ubiquity of the domestic canine across the tapestry of human civilization, our comprehension of its genomic intricacies has hitherto been lamentably circumscribed. The magnum opus, christened as the Dog10K Initiative, under the aegis of luminaries such as Dr. Jeff Kidd of the University of Michigan, Dr. Jennifer R. S. Meadows of Uppsala University, Sweden, and Dr. Elaine A. Ostrander of the NIH National Human Genome Research Institute heralds a new age in canid genomics. Methodological exegesis and revelatory findings. Employing the prodigious computational acumen of the Great Lakes supercomputing coterie at the University of Michigan, the consortium embarked on a meticulous genomic odyssey. They sequenced and dissected nearly 2,000 canid genomes, spanning a kaleidoscope of 321 breeds and wild specimens, juxtaposed against the referential genome of a German shepherd, christened Mishka. The analytical sojourn unraveled a staggering 3 million single nucleotide polymorphisms, SNPs, within each breed, quintessential fulcrums underpinning genetic variegation. Furthermore, an intricate genomic tableau was unveiled, with sequences evanescing and materializing in juxtaposition with Mishka's referential DNA. Morphological genomic elucidations and retrogenic phenomena. Dr. Kidd, in an epiphanic elucidation, expounded that the canine taxa could be perspicuously stratified into circa 25 salient clades, harmonizing with extant taxonomies predicated on morphological paradigms. Ergo, morphological idiosyncrasies emerge as the paramount vectors modulating genomic heterogeneity. An equally arresting revelation was the pronounced proliferation of retrogenes, 926, to be precise. Notably, the FGF4 retrogene manifests conspicuously, engendering the diminutive extremities emblematic of breeds such as dachshunds and corgis. These retrogenic apparitions could be a testament to anthropogenic selective proclivities, swayed perhaps by esoteric aesthetic dogmas, conjectures Kidd. Portending implications in the horizon of canine genomic prognostications. The voluminous data cache proffered by the Dog10K consortium unfurls a veritable Pandora's box of investigative avenues. Beyond the elucidation of canine morphological diversification, the genomic troves beckon researchers to probe into the genetic substrates undergirding prevalent canine maladies, most notably oncological predilections. The Dog10K consortium's magnum opus transcends the realms of academic esoterica, poised to revolutionize our understanding of an emblematic companion species. This magisterial work augments our grasp on the labyrinthine genetic mechanisms bespeaking disease susceptibilities, heralding novel therapeutic stratagems.